Hello, my name is Sotirios Pistas. I'm a consultant neuroradiologist at the National Hospital for Neurology and Neurosurgery. Gliomas consist of a very heterogeneous tumor group, and there are several underlying pathological mechanisms that are influencing the survival rates of the patient. One of these mechanisms with major prognostic and diagnostic consequences is the mutation of the IDH, which stands for the isocitrate dehydrogenase. The IDH mutation leads into the accumulation of the oncometabolite 2-AG, 2-hydroxyglutarate, which actually decreases the survival rate of the patient and the response rate to the standard therapy, which is chemoradiation accompanied by surgery. Our impact on the clinical routine and the patient prognosis would be to provide a non-invasive pharmacodynamic biomarker allowing us to assess the mutation status of IDH. This has implication for the patient stratification according to the best available treatment and will improve overall the clinical management of patients with gliomas. Now I would like to introduce you to Enrico, who is my colleague responsible for the technical implementation of the MR spectroscopy in the clinical routine.
My name is Enrico De Vita. I'm a physicist. I'm a senior lecturer at the Institute of Neurology, and I'm a clinical scientist for the Neuroradiology department of the National Hospital for Neurology and Neurosurgery. Until recently, it was only possible to detect the IDH mutation by invasive biopsy of the tumor tissue. MR spectroscopy gives us a completely non-invasive tool to measure whether we have an IDH mutation in the tumor. Besides diagnosing the presence of IDH mutation, it would be really useful to follow the tumor serially during therapy and to find out whether the tumor is responding to therapy. In order to do this properly, we have to be able to have a technique that is able to sample the concentration of 2-Hg or the whole tumor volume in 3D. We have just set up a collaboration with the Massachusetts General Hospital that have recently proposed a technique to be able to sample in only 10 minutes in 3D the concentration of 2-Hg in the whole of the brain tumor. Moreover, they have shown that the technique is reliable and reproducible over serial scans. In this PhD project, you, together with us, will be able to test locally the ability of this new sequence to detect the presence of IDH mutation completely non-invasively. The methodology will support clinicians in designing personalized treatment plans, and once clinically validated, the long-term goal is to be able to follow up tumor treatment without the invasive biopsy. Here in Queen Square, with the partnership between the UCL researchers and the National Hospital for Neurology and Neurosurgery, we have an amazing opportunity to be able to advance patient care.